Hi guys, welcome back to Blue Tongue TV. Today what we're going to look at is pairing a recessive trait to an incomplete dominant trait. And to do that we're going to actually use albinism and hypermelanism. So when you actually pair these two together you get a full litter of what's called double heterozygous animals. And the whole litter will look like that. So the recessive trait, as we've covered, you can't see, but the hypermelanistic trait, incomplete dominant trait, you can see in this animal. So the whole litter is going to look like the incomplete dominant hypermelanistic animals, but carry a copy of the recessive albinism gene. You'll remember in episode 1 when we looked at recessive genetics that there was 4 animals in the ratio that could come out when pairing heterozygous animals, and that was homozygous recessive, 2 heterozygous and 1 homozygous wild type. And that was also the same in episode 2 when we paired the heterozygous form of the hypermelanistic animals. In that ratio what came out was homozygous hypermelanistic, 2 incomplete dominant heterozygous animals and 1 wild type animal. So you have 4 in each ratio. So when you overlay the 2 together, which you're doing when you're creating double genetic animals, you're then timesing your 4 in one ratio by the 4 in the other ratio, which will give you an entirely different ratio, and that's 4 by 4, which is 16. So when we pair the double heterozygous animals, you're going to get 6 different phenotypes, which are as follows. We've got hyperhet in appearance, normal wild type in appearance, normal albino in appearance, then we've got high orange albino in appearance, which is your albino het hyper, homozygous hypermelanistic in appearance, and then finally number 6, which is your double homozygous hypermelanistic albino, or your larva. Then we've got 9 different genotypes. You've got normal wild type, heterozygous albino, then we've got double heterozygous albino and hypermelanistic, and also single copy heterozygous hypermelanistic. Then we've got recessive albino. Then we've got albino het hyper. Straightforward normal hyper. And then we've also got hyper that's het for albino. And then finally your 9th different genotype, which is the ultimate, again, your larva, which is your hypermelanistic albino. So then when we pair some of the resultant animals from the double het pairings, larvas are going to come out in the following ratios. We said from double hets you're going to get larva as 1 in 16. But then when you pair a double het to an albino het hyper, you're actually going to get 1 in 8 larvae. Then again, when you pair your double heterozygous animal to your hypermelanistic het albino, you're going to get 1 in 8 larvae. If you pair your hypermelanistic het albino animal to your albino het hyper animal, you're going to get 1 in 4 larvae. Then when we start using the larva itself, if you pair that to a double heterozygous animal, you're going to get 1 in 4 larvae. Paired to a hypermelanistic het albino, you're going to get 1 in 2 larvae. Paired to an albino het hypermelanistic animal, you're going to get 1 in 2 larvae. And then finally, the ultimate, when you pair larva to larva, you're going to get 1 full litter of larvas. You could say then, with a full litter of larvas, that the volcano has well and truly erupted. It's time for the classroom again, and Dr. Steve.
Hey guys, Steve here. Today I'll be showing you how to use Punnett squares for double genetic animals. The example we will use is the larva bluetongue. The larva is homozygous for two traits, albinism and hypomelanism. Albinism is a recessive trait, with the hets being indistinguishable from the homozygote normal. We will use big A, small a as the two alleles. Hypomelanism is an incomplete dominant trait, with the hets usually being identifiable as they are often darker than homozygous normal. We will use a B as the wild-type allele and a C as the hypomelanism allele. For our Punnett square, we will use parents which are heterozygous for both albinism and hypomelanism. The genotype will be big A, small a, B, C. Each parent passes on one allele from each gene onto their offspring. You can see that there are multiple combinations. Big A, B, big A, C, small a, B, and small a, C. It is these combinations that we write along the top and side of the Punnett square for each parent. Like any other Punnett square, the alleles from the mother are transferred to the boxes below, and the father to the right, filling 16 squares representing the offspring. In the offspring, there are 9 different possible genotypes. The normal wild type, het for hypomelanism, het for albino, hypomelanistic, double het, which will be 1/4 of the litter, het for albino hypomelanistic, albino, albino het for hypomelanism, and the double recessive larva, which is a 1 in 16. Because we can't identify the young that are het for albinism, but can usually identify hypomelanistic hets, there are 6 different possible phenotypes in the litter. The normal wild type, which you can see 2/3 of, are hets. The wild type het for hypomelanism, which are darker than the normal. 4/6 of these are het for albino. The hypomelanistics, 2/3 are het for albino. The normal albino, the albinos that are het for hypomelanism, which typically show higher amounts of orange than normal, and our double recessive larva. Now I have demonstrated the 16 square Punnett square, try working out your own using parents with different genotypes. Thanks for tuning in guys. Be sure to watch next week where Joe and I will show you how to breed a snow bluetongue.